of genetic engineering. Our next topic is going to be cloning. So far we've talked about gel electrophoresis, and we've also talked about genetic engineering. Next up, we're going to take a look at cloning. So before we get into the steps of cloning, let's take a look at a definition of what exactly cloning is. So cloning is the production of a group of genetically identical organisms, or a group of cells artificially derived from a single parent. Today we're going to be talking about specifically two types of cloning. The first one, reproductive cloning, and the second one, therapeutic cloning. We are first going to take a look at reproductive cloning. So let's jump on in. What is reproductive cloning? Reproductive cloning is the deliberate production of genetically identical individuals. Each newly produced individual is a clone of the original. So we're making a direct copy or replicate of the original. You may have heard in other science classes the sheep dolly. Dolly was the first ever organism, the first sheep to be cloned from an original parent sheep. So this sheep dolly had the same exact DNA as the original sheep it came from. So how exactly did scientists do this? Let's go step by step and take a look at how this happened. Step one. From the original donor sheep to be cloned, a somatic cell, a non-gamete cell, from the udder was collected and cultured. The nucleus was removed from that cultured cell. So let's take a look at the image. Here's our donor sheep. This sheep gave a nucleus that's going to be used further into step two. Looking at step two, an unfertilized egg was collected from another sheep, let's say sheep 2, and its nucleus was removed. So let's look at the image. Here we have sheep 2. This sheep we got an egg cell from. However, the nucleus of this egg cell is going to be taken out and used. Let's now take a look at step 3. The nucleus from the udder cell, so the nucleus from this sheep 1, was put into the egg cell from sheep 2, this one. So you can see here in the image that the nucleus from sheep 1, or the all-white sheep, was put into the empty egg cell of sheep 2. So when we do that, we are on to step 4. The new cell developed in vitro into a zygote and into an embryo. So here we have the fused cell. We have a nucleus from the sheep 1, and we have an empty egg cell from sheep 2. When those fuse, as it's developing and as it's differentiating, it's going to turn into an embryo. Okay. This embryo now, we're going to look at step 5. The embryo was placed in the womb of a surrogate sheep. So now we're going to put the embryo that was created from the nucleus of sheep number 1, and we're going to put it into a foster mother. So we're now at sheep 3. We're going to put it into a foster mother. When that embryo gets put into the foster mother, the embryo is going to grow even more, and Dolly will be born. Okay, the first sheep ever cloned. So again, we're taking the nucleus of sheep number 1, an empty egg cell of sheep number 2, and allowing them to fuse together. So we're using the DNA from sheep 1 to create an embryo. And that embryo is going to grow into a little lamb and be born. This lamb has the same DNA as sheep number 1. Very, very fascinating stuff. Very interesting. What can also help you with looking at reproductive cloning is this video that's linked in the slides. If you want to take a look at how exactly the first cloned sheep dolly was made or produced, you can go and take a look at this video. So that is reproductive cloning. The next kind of cloning is therapeutic cloning. Now this is a little different. Let's take a look. Okay, therapeutic cloning is not making cloned organisms like we were in reproductive cloning, but rather here we're going to clone cells. Now, how are we going to do this? Here's another process that we can go step by step, taking a look at the step and then taking a look at the image. So, step number 1. The nucleus from a skin cell of a diabetic patient is removed. So again, remember, we're cloning cells here, not organisms for therapeutic cloning. So again, step number 1, the nucleus from a skin cell of a diabetic patient is removed. So looking at the image, we have it here. The nucleus from a skin cell is being removed. So we're taking the nucleus, we're taking that DNA. Step number 2, the nucleus is removed from a human egg cell. So now taking a look at step 2, we are now going to have an empty egg cell and a nucleus from the patient, from the person. Okay. Now let's take a look at step 3. The skin cell nucleus is inserted into the denucleated human egg cell. So now we're here. The nucleus from the person is going to be inserted into that empty egg cell that we got from step number 2. Now let's take a look at step number 4. The cell divides and replicates and begins to develop. So we have it here, the nucleus is in the egg. It is now going to divide and develop and grow and become a group of cells. Okay. Looking at step number 5, the embryo reaches blastocyst stage. So now we have a group of cells all together that stemmed from this original cell that has the nucleus from the patient. Taking a look at step 6, the embryonic cells are grown in culture. So looking at the image, we now have this blastocyst with many cells that came from the patient, and scientists are going to allow it to grow in a petri dish. Looking at step number 7, the stem cells differentiate into healthy pancreatic cells. So scientists were able to manipulate these cells that are stem cells, and they were able to manipulate them into creating healthy pancreatic cells. Now remember, our patient from step number 1 is a diabetic patient. That means that something isn't going correctly in insulin production. So if we create healthy pancreatic cells, these healthy pancreatic cells can help us with forming or creating the right amounts of insulin that this patient might need to help with blood glucose levels. So that was step number 7. And then looking at step number 8, those now differentiated pancreatic cells are going to be inserted back into the diabetic patient. So this was able to create cells that the diabetic patient needed. So again, if you would like to look step by step into what was going on with therapeutic cloning, you can go through the next following slide. So looking at slide 20, you can go from step 1, 2, 3, 4 to see what's going on. In slide 21, you can go down from 4, 5, and 6. Remember to go this way because we're looking at therapeutic cloning. And slide 22 allows you to see, okay, once we're done with step 5, how do we go from the petri dish into pancreatic cells and then being able to insert it back into the diabetic patient, which will be helpful for them. So we took a look at therapeutic cloning, but therapeutic cloning has some ethical concerns behind it. So let's take a look at what these ethical concerns are. So because therapeutic cloning starts with the production of a human embryo, it raises fundamental issues of right versus wrong. So the question is it really ethically acceptable to generate a new human embryo for the sole purpose of medical research? In nature, embryos are created only for reproduction, and many people believe that using them for experiments is unnatural and wrong. So there is an ethical debate behind therapeutic cloning, but it is still a very interesting topic to discuss because it is such a new new topic in science that there's a lot of discussion and a lot of talk going on about it. So I hope you enjoyed this video. Please feel free to ask any questions during the Google Hangouts, or feel free to email as well. And thank you. Have a great rest of